So here's where we were. We were saying this abstraction of a neuron is valid, at least for one type of neuron, a big, beautiful Purkinje cell. This type of model seems to be a better model for neurons with long, that are different from terrestrial trees. Every time you look at a tree now, I want you to think about this, how most terrestrial trees seem to have this Purkinje cell-like shape, where most of the stuff is on little twigs, whereas pyramidal neurons, especially human ones, most of the stuff is on, there's a little bit of branching and then these long unbranched sections which create kind of little subunits. And so for those types of cells, a two-layer model is more appropriate where you think, and where they kind of act independently of each other, the non-linearity acts independently on this stuff in this dendrite, and then it adds up with the stuff in the output of that dendrite, and that's the output of a neuron.
So yes. I was wondering what actually makes this compartmental nature of the dendrite. You say it's the sort of non-branching, but if you have a branching one, it should be the same story basically, right? Isn't it the NMDA spikes that actually do this, or the dynamics, or is it really the non-branching that makes this subunit?
No, it's not. If you could have very branched things with NMDA channels, and there will be local spikes generated. Yeah. It's just somehow the logic of these cells seems to be to put a set of inputs onto this dendrite, which will interact closely and under the same nonlinearity, and different from the inputs to a different dendrite. I'm not saying that it has to be long unbranched sections. I'm just saying that they happen to be, in the case of pyramidal neurons.
But what is sort of what's the cause for?
Why would it want to be that way?
No, I mean, what's the real reason? I mean, you say you observe these neurons, and they are somewhat, in some sense, they build compartments. But what's really the reason, the functional reason for this compartmentalization, whatever?
Are you saying what is the computational benefit of having multiple compartments?
No, I got that. But what's really the? I mean, you say if it's a branch dendrite, could still be compartments, right? Could still have.
Well, this is really
could still be a valid model for a branch, for a neuron with branches. So it doesn't seem to be this long unbranched parts of the dendrite that make this a reasonable model.
Okay. Let me just make a relatively weak statement so that it doesn't bother you. Pyramidal neuron dendrites are largely, are often characterized by tending to have long unbranched sections. They will be like kind of functional subunits. Other architectures are possible. Okay. Now here's a problem with this two-layer model. Remember, well, let me see if I come up here. Oh, wait, here. This says that there is a sigmoid in this dendrite. It's a sigmoidal input-output function. Now, is that a valid abstraction? And it says that, by the way, that the inputs to a dendrite add, and it doesn't matter where they are. They're just a big, it's a big collector, and in that dendrite, and the inputs kind of add up, and then they go into the sigmoid. So there's no sense of a location, like a synapse being at this location versus that location having a different effect. So let's see about the validity of that. So this was an experiment done. There's a recording at the cell body. These are the voltage traces that are being recorded. And there was ionophoresis of glutamate in these experiments. So glutamate's being kind of squirted out onto these dendrites to stimulate them, to stimulate the glutamatergic synapses at different places. But at one place, let's say that corresponds to this place, the stimulation is turned up and turned up and turned up, and it's kind of creating a series of curves like you saw before with the uncaging. This is ionophoresis. And here's a spike that's generated. And if you were to do the same thing closer into the cell body, then you get this trace. Now, the reason is it's because the same thing is basically happening at that point in the dendrite, but because you did it closer, there's less attenuation of the voltage moving in towards the cell body, so you see a bigger signal. And if you did it farther away, you would see sort of this kind of a pattern with little subthreshold things, and a spike is generated, but the spike looks really tiny that far out. Not because the spike is tiny out where it is, but because the voltage attenuates a lot on the way in. And if you kind of generated input-output curves, like the amount of input, and then you plotted it against the voltage that occurred, the peak voltage that occurred, you'd get something like this. You'd get a low threshold low amplitude sigmoid for this position, medium and large sigmoid for a different position. So even within one dendrite, you've got 3 sigmoids that have varying thresholds and amplitudes, which is not part of the paradigm of a typical like neural network unit. So that's already something that we have to contend with. So the question that it raises is, okay, that's already kind of breaks our paradigm, but then let's go one step. Let's do, what's the next experiment you want to do? You want to take 2 inputs and you want to vary their locations and look at how they combine. So you could, one thing we could ask is, let's just consider 2 locations, and I call one a driver and a modulator, or I can flip it. It doesn't really matter. That's a semantic term here. And we're just interested in what is the symmetry or asymmetry between a proximal and a distal location on one dendrite. We know that they will individually behave differently, but what is the symmetry or asymmetry of the interaction between those 2 inputs when you apply them together? And it could be that you could have an input-output curve of the driver, and then as the modulator is increased, it could do something like this. It could lower the threshold without changing the amplitude. It could change the gain, essentially, but without changing the threshold. And it could do some kind of a combination of those. I mean, we don't know. So this is the question to ask. What about multiple inputs that are spatially delocalized or spatially separated? So there were a couple of papers. We collaborated with Jackie Schiller again. Some of my former students, Bardia Behabadi, Manuka Jadi, worked on these modeling aspect of it to kind of look at this question in a joint experimental and modeling context. And here were the experiments were kind of like we saw before, where there was uncaging or uncaging or electrical stimulation at 2 sites on a real dendrite, and there was a recording made at the cell body. And then the electrodes would be activated at different levels. Well, actually, the top one was meant to be a model, and the bottom one was meant to be the experiment. And here you can see an input-output curve. Different levels of activity of the distal input eventually generates a little spike at a certain threshold level of intensity here in an experiment, and there in a model. And then if that curve is repeated in the context of a proximal input that is increased to generate, and then a new curve is generated, and then it's increased again, and a new curve is generated, what you see in the model is that there's a left shifting of the threshold of the sigmoid and an increase in the excursion, an increase in this sort of takeoff point. So it's sort of a characteristic progression of these sigmoidal input-output curves generated by the distal input in the context of an ever larger proximal bias or a proximal modulating input. And in the experiments, it was very similar. The distal input by itself would generate a little sigmoid, and then the sigmoids sort of shifted to the left and the spikes became bigger in excursion and sort of having a longer or higher takeoff point. So it was a very similar pattern. And then so the effect of a proximal modulator is to sort of modulate the input-output curve of the other input of a distal input in that particular way. And then if you kind of do the opposite experiment, and you use this one as the driver, and then you generate an input-output curve, and then you do it in the presence of an ever larger distal input to generate a series of curves, then you get a very different pattern. You get this pattern where there's a left shifting of the sigmoid with no change in the amplitude, and eventually kind of a linearization of the input-output curve. And that's what happens here. There's a left shifting of the input-output curve with, well, maybe a slight degradation in the amplitude, but then eventually this linearization occurs. And so that's the effect of a distal modulator on a proximal input is no increase in the excursion size, but a left shifting of the sigmoid. So it's an interesting and strange asymmetry that all of the data together, comparing model and data, sort of said the same thing. So actually, here, let me just go to here. So here's a plot of that. If you do this in a model and you take a proximal and a distal input, you plot proximal input there, distal input there, and you plot the response at the cell body, what you see is a two-dimensional asymmetric sigmoidal function. That's what the dendrite does when you put 2 inputs in that are not at the same place. That is not any simple generalization of a 1D sigmoid, is it? It's a 2D nonlinearity. It depends on the location of the input. interestingly, and I think this is I think this should be compelling to you, many of you, in terms of people who like to make models of things. This surface is produced by this circuit, which has 5 conductances in it. This model has, a full compartmental model, thousands, I don't know how many, 443 compartments. Each compartment would have many differential equations. They're all coupled, it's very nonlinear. There's NMDA channels, AMPA channels, cable properties, etc., morphology, and that produces this input-output curve, and this is produced by 5 conductances with no time at all. This is all steady state. So it's a virtually perfect reproduction of what one thin dendrite does with NMDA channels for delocalized inputs. It produces an asymmetric 2D sigmoid. Now, does anybody think that's weird? You do?
Yeah.
Yeah, it's kind of interesting. It's like, what the heck is a 2D sigmoid for? It's asymmetric. But what I like about this is that this extremely simple model captures the behavior of a very complicated model very well in that respect. So we also took this, now maybe this is an artifact of, so real neurons actually generate action potentials, not just 10 millivolt excursions from resting potential. So what about when this thing is taken up to a range of intensity of stimulation so that the cell is firing action potentials? Shall I pause before I finish my sentence, or do you want to? Well, just to say it's the same, it's a very similar pattern when the cell's producing action potentials. Here's the distal input by itself produces, so now the 2 inputs are bunches of synapses being activated by trains of action potentials, and they're, so the synapses are being repeatedly activated. There's noise being injected to the cell body so that sort of simulate in vivo-like conditions, and then the membrane potential is measured at the cell body, and what the output is now is not how much voltage is caused, it's how many spikes per second are caused, and that's plotted on this axis here. But again, you have a little sigmoid caused by the distal input, a big sigmoid caused by the proximal input, and this kind of smushy asymmetric sigmoidal thing in between. And as before, the effect of the proximal modulator is to sort of left shift the threshold and increase the excursion, and the effect of a distal modulator is to left shift the threshold and with no increase in the excursion, and that's the same asymmetry that we saw before. Yes, Murray.
So when you have 2 inputs on a dendrite like that and they're coactive, wouldn't you expect the more distal input to be more affected because its current would be shunted on the way to the cell body because of the synaptic conductance of the more proximal input, and it wouldn't work the other way around.
You're saying the distal input should be more effective?
No, I'm saying that the effect of having 2 inputs activated at the same time is you get a conductance at both points, and the current flow from the nearer one is not going to be so affected by the more distal input, but the more distal input will be shunted more because of
but shunting suggests a reduction of, but actually there's a synergy because these are NMDA channels, and they.
So I'm saying both. I can understand the synergy, but I would have thought the conductance would have a bigger effect on a more distal input.
Well, this is. Let me escape from this by saying that. Those kind of word pictures, the ones that you're trying to construct and that we tried to construct for months and months and still do, are very difficult to make work. It's very difficult to take to put into words all of the things that go on inside a cable with NMDA channels in a way that you'll remember for more than 3 seconds and be able to use. So the way that I boil this down in my mind is I say, if you were to have 2 inputs to a dendrite that are NMDA channel that activate NMDA conductances, and you have a cable there, then you're going to get an asymmetric 2 sigmoidal relationship. And I kind of understand. I understand this curve. I understand this curve. And what happens in between is kind of smooth. And that's about as far as my intuition can carry me. We can sometimes get a little bit more insight into a little, if we ask a very specific question. But in terms of kind of breaking it into, reducing it. In terms of simple concepts, is very difficult, and when inhibition comes in, it becomes even harder. I mean, let me show you why it could be. So, these are examples of like, okay, let's go to the simulation and let's take one of these asymmetric 2D surfaces and start playing with some different parameters. We could have different separations. We could have synaptic facility. We could model synapses that have slightly different properties that facilitate. We could have spines with different neck resistances. The point is that the biological parameters that are available to the neuron provide for a rich set of these nonlinear, asymmetric, multidimensional sigmoidal interaction functions that can be made, okay? And that's what the hardware can do and does do, even just for 2 inputs. And here's a case where even the synapses that are moving along this axis don't even have NMDA channels. So if you take those out, you can nonetheless get an input-output curve that is linear, but whose gain is increased by the other modulatory input, which does have NMDA channels. So there's just a lot of different possibilities, and that's kind of what we see our job as doing a little bit, is to sort of work out what the parameters that actually exist in the biology and in these cells make available to you, the brain designer out there, who is trying to use this kind of biophysical mechanisms in order to compute something. There's a lot of flexibility. So inhibition, people have asked. So it turns out it's maybe a lucky thing, but if you were to have another input, 2 inputs, and one of them is an inhibitory input, it turns out to have a kind of a similar but anti-parallel effect as excitation. So that whereas this is a case of proximal inhibition, what it does is it has a significant, there's a slight elevation of the threshold and a significant depression of the excursion. And distal inhibition has the effect of raising the threshold without changing the excursion. So it's kind of doing the opposite of what the excitation was doing in that place. And the same for in a model. So to understand the combination, to understand excitation and inhibition both, you can kind of think about them. And as I say this, I'm very cautious to say this because it probably won't turn out to be true in all cases, and I don't know how general this will turn out to be. But anyway, in at least the simple cases that we looked at, there seemed to be kind of an opposite opponency to this, where if you were to put an excitatory modulator on distally, you're driving it with this black input here, and then you use this as a distal. What we expect this to do is to lower the threshold, and it does. It lowers the threshold without changing the excursion. And then if you just put an inhibitory synapse there, it kind of does the opposite. Maybe not in equal amounts, but it does the opposite. If you do combine them together, then they kind of cancel. So it's creating a little bit of a sense that there's an arithmetic that isn't crazy between excitation and inhibition. They kind of both obey the same location-dependent rules, and they kind of cancel out each other's effects, or they go in opposite directions. To your point of like, how do you do positive and negative things and so on. These are opposite direction modulatory effects. This is the same sort of a deal where a proximal modulator, if excitatory, does this kind of a shift to an input-output curve, and a proximal modulator that is inhibitory sort of pushes you lower in amplitude and further to the right. These two didn't perfectly cancel in this case, but they kind of sort of canceled if you put them both on at the same time. So there's some logic to this. But you can sort of now see that the possibilities for using excitation inhibition in different locations to modulate responses nonlinearly are wonderfully rich.
So this is a cartoon of experimental data, right?
No, this is a cartoon from a model, but it summarizes the experiments.
Model.
This model may have been done in MATLAB using just representing the NMDA channels in a simple cable.
Okay.
I can't remember what Monica did on this particular one. So now, I'm trying to put into words what I just said in terms of what opportunities this biophysical mechanism of having a dendrite that's nonlinear, cable, NMDA channels, excitation inhibition that can go to different places. The opportunities that provides to the cortex to do things to the neuron, what opportunities are there? Well, a neuron that is going to affect this neuron with some kind of a modulatory input now has the choice of steering its contacts closer to the cell body, farther from the cell body, uniformly, whatever it wants to do. It has the opportunity to do that, And this is done, as Murray will tell you, like in the thalamus, there's going to be inputs that target distally. Well, actually, in almost any neuron, there are neurons that have chosen to target distally, proximally, this, here, or there. That is a capability that the brain has to steer inputs proximally and distally. And by doing so, these modulatory effects can be steered or tailored. And the inhibition, so this is interesting because the inhibition is different where there are dedicated neurons which target proximal and distal locations, but then the pathway can choose the degree to which it activates one of those to affect the amount of proximal versus distal inhibition that will be applied to the neuron. And I want to show one example of this. There's a lot of kind of excitement in recent years as the ability to label different types of neurons has increased with different kind of molecular markers, genetic, viruses and so on, can light up different classes of interneurons. So here's a pyramidal neuron. This is a parvalbumin containing inhibitory interneuron, which tend to target the cell body of pyramidal neurons. This is a somatostatin containing neuron, which tend to target the dendrites of pyramidal neurons and tend to inhibit PV neurons. So there are these circuits, these circuit motifs like this, which have been proven, shown, demonstrated, tested, et cetera. And when you look at this and you think about what I've just been talking about, these proximal distal interaction effects, you now see that, well, okay, a modulatory, here's a neuron which is off, outside, let's call it the receptive field of this neuron, but its axons run over and make contact. By steering a contact to different distances from the soma, can have very different effects, from gain modulating effects to threshold modulating effects. Similarly, that by activating, and these contacts do make contact onto these cells, they do make contact onto these interneurons. These interneurons they inhibit the inhibitors. And so actually, if you look at the logic of this, a somatostatin neuron, by being excited, delivers inhibition here, and it also reduces the amount of inhibition here. So what you do by activating a somatostatin neuron is you take the inhibition, which was mostly at the soma, and you shove it mostly to the dendrite. So what does that do? So we actually simulated that case to see what that did, and we kind of had an idea. We followed the logic that we had built up from these biophysical kind of studies. And if we just sort of erased the whole inhibitory interneuron circuit, and we just considered this modulator affecting this and this driver and this modulator, and the driver's distal and the modulator's proximal, then we get one of those curves, those 2D surfaces like I showed you before. And if you slice it up, you get curves like this with that characteristic sort of left shifting and steepening of the input and output curve. But if you put the circuit in place, these two weird things happen, which is that as the modulator increases, it keeps putting out more and more distal inhibition, which pushes these curves progressively. Each one, this one goes further to the right, this is a little less far, and so on. And it tends to line up the curves with each other. And then because you're progressively relieving the cell body of inhibition, you have a lot of suppression on the first curve and not very much suppression on the last curve. So you end up with this kind of high dynamic range, multiplicative scaling of the input-output curve. So in other words, this curve looks like a small version of, like this curve is like 3 times the value of that curve. So it takes a kind of a complicated looking pattern and it turns it into a simpler looking multiplicative scaling. So we don't know if this is what that circuit is really for, but it kind of, it's possible that one of the uses, or one of the ways in which the cortex uses these types of local circuits, is to just exactly tailor the modulatory effect to be something other than, an excitatory only effect would look like this, and one that includes these inhibitory motifs would be of a different nature. And we just kind of explored whether multiplicative scaling, which is a very common operation in many computations, would possibly be the result, and we found out that that was a very convenient thing for this kind of a circuit to do. Yes?
What's the y-axis in those points?
Oh, response, like spikes per second. I'm sorry, that should have been.
How would this be affected if spike timing were to be taken into account?
Well, that's a good question, but you remember that I said that we were kind of looking at a rate version of this. the minute you introduce spike timing, I mean, anything can be different. I mean, you could have spikes that don't overlap in time with each other, and there's no interaction of anything to anything. So we'd have to, when you want to deal with spike timing questions, you have to kind of be very specific about what scenario exactly are you talking about, because it will matter. Yes?
So, to what degree do the details matter for this effect? Because, you know, Chance and Abbott showed in 2000 that having kind of a combined excitation with inhibition, you get multiplicative gain modulation. So, I guess, you know, if you start just, you know, taking apart the components of this circuit, the sum, the PV, and so on and so forth, which one is actually critical for the multiplicative effect?
Well, multiplicative, making this curve be a scaled version of this curve, you can sort of see intuitively, you kind of need to line up the thresholds, so you need to push them ever closer to the right, and you need to have, and then you'd like to have as large a kind of a dynamic range as you could get. So this is by no means the only way that you could get a gain effect in a neuron. You know, the apical tuft is a way to do it. Chance and Abbott's proposal is a way to do it. There they were basically just turning up an inhibitory conductance and there was noise and they did it. So and that didn't have anything to do with dendrites at all. In fact, they were point neurons, if I remember. So this is just pointing out that a modulatory input in the cortex has a lot of flexibility, including this effect, but including a large spectrum of other cases as needed. Yes?
There's another way of looking at this circuit. One takes into account that there are so much fewer parvalbumin and so much total neuron compared with their 2-3 pyramids. So when you look at many of these circuits, you know, they must share this parvalbumin neurons, right? So then you have a higher dimensional computation and new effects taking place, right? Rather than, you know, looking at just this local projection that you consider in these curves here.
Yeah, I mean, so what you're saying is that, Wolfgang, is that this cell will target multiple cells and this cell will target multiple cells, and so it's not like this is a dedicated interneuron in this circuit. Yes, that's absolutely true.
Sorry, just to build up on what Wolfgang pointed out, of course that has lots of implications for distributed replications in terms of like the noise correlations across multiple neurons. I think that's a very interesting kind of thing to come.
Yes, and so this is by no means an explanation of how cortical circuits work. It's what we're trying to do is trying to identify the parameters available to neurons and the neurons next to those neurons to affect each other, you know, so that we can understand, like, how do you build up computations with this kind of hardware. And we just kind of, we're really in a, you know, when you start to get a circuit this complicated, but we're not modeling these neurons in a sophisticated way. If we did it in a sophisticated way, things might change. You know, it's just, it's kind of scary. There's a lot of flexibility here. But then again, you know, we have to find the sources of flexibility. If you think about the cortex, this, you know, if most of us looked under the microscope at a piece of cortex and just picked it out of the brain at a random, you wouldn't know where you were looking because it all looks pretty similar. I mean, you know, yes, there are some differences that even, you know, I might see as a non-trained neuroanatomist, but it's kind of an architecture that has been conserved and is used and has some parameters changed. But those parameters can be changed so that that machine can process visual images, process sounds, process whisker inputs which come in over time, process tastes, produce language, produce thought, produce planning, you know, I mean, produce emotions. This is one basic architecture whose parameters can be changed to do all of those things better than we know how to do any of them. So there's got to be some flexibility there. And that's what we're trying to do, is sort of figure out where, you know, where it's hiding. And here is a place where some flexibility exists to tailor the amount of excitation that goes here and there, to tailor the amount of inhibition that goes here and here, etc. This is interesting. You probably, nobody seems to know this. I found this, and I'm bragging like I found it. No, well, I read the paper, but they found it. In primates, there is a system of inhibitory interneurons that is extremely spatially localized. It's very numerous. This is the top, so this is the cortex, and these inhibitory neurons, they're shown here, they're called double bouquet cells. They have these very narrow columnar axons that are like a little 10 micron sort of cylinder, which is a small, I mean, this is like maybe 100 microns or something, 150 microns. This is a 10 micron cylinder that goes up and down, makes inhibitory contacts onto one little bit of a dendrite. And then there's another. Then you move over 20 microns and you get another one. This is a top view of the columns, and this is the side view of the columns of these axons. So they produce this kind of a grid of inhibition that targets everything that it targets in a localized way. So there's localized inhibition happening on different dendrites. Imagine that you're sticking out your branches like this, and you're going through these kind of columns of light or something, and you cut into them at different places depending on which dendrite goes where. And so you're receiving focal inhibition at different points on different dendrites, and maybe this is a way of producing diversity so that different dendrites can act like can have different sorts of I think this is actually that I didn't plug my computer in. So I hope that it doesn't need much more than just me pushing a button. Give it a second. I'll extemporize here for a second. This grid produces a very, very intricate pattern of inhibition in the cortex, and nobody knows what that's for. But knowing that there are spatial integration effects that are nonlinear and location dependent makes that a significant fact.
So about the grid, it sounds like the double bouquet cells are going vertically. You said something else is going horizontally? Or is that, did I mishear that?
Well, if you take a pyramidal neuron, the great majority of the contacts that land onto a pyramidal neuron are not vertical axons coming to drive the cell with sensory information, let's say in the sensory area. They are horizontal connections coming from other pyramidal neurons, something like 70%. So the cortex seems to be designed to provide the opportunities for extremely intricate lateral modulatory effects where cells interact nonlinearly with each other or they affect each other. Oh my God, it's perfect. The panic that, thank you, Apple and whoever else. Yes.
So does the inhibitory neuron span multiple layers within a cortical column?
Say that one more time.
Oh, so does the inhibitory neuron, does that span multiple layers of cortical column?
Well, you can see here that that it's traveling vertically. I can't remember exactly whether they go from the superficial layers to the deep layers. I think they don't, but just here, De Felipe et al. 2008. I'll send you the reference. It's just a little known fact about about this grid. That's again, it's kind of grid. When you see grid-like things, the engineer inside you goes, "Oh my God, that's something that I could probably understand." Well, we hope so. Yes.
So yesterday came up the question whether we need these different types of neurons, so the different morphology, primarily to implement different types of connectivity rather than different functions. So this would be a neuron that has a very specific morphology, so to induce a particular kind of connectivity. So what's your stand on this? I mean, would you say that inhibitory neurons might, in terms of function, might be similar, but you need different types in order to have different connectivity?
Yeah, I mean, it sure looks like it. I mean, inhibitory neurons can have more than just, they can be different from each other more than just how do they target their targets. Do they target them like basket cells cover the cell body with inhibitory contacts in a basket, and somatostatin cells kind of sprinkle the inputs more distally. These cells hit a column. Yeah, so those, but it could be that the actual synapses themselves behave similarly. They release GABA, they activate similar conductances, So yes, morphology and the targeting pattern is a major differentiator between inhibitory neurons, and it's part of what gives the circuit its personality. The difference between a V1 and a primary, like a barrel cortex where whisker input is coming in, which is a totally different signal. I mean, you can imagine something so different as that, or sound from images. You're going to get the tailoring of the circuit is going to be some of it's going to be which inhibitory neurons are being recruited in which ways to do what things, and that's part of the flexibility of this system. So just to summarize that is to say that, okay, we still think this is a two-layer model, but the subunits are more complex. They're more elaborate. They have these nonlinear spatial, intrinsic spatial processing capabilities, analog processing capabilities. And I'm representing that as a little little 2D sigmoid there. So it's kind of departing from a conventional neural network in that sense. But although we're still talking about a cell body that's summing some of them together. Yes?
So right now you talked about two inputs to the dendrite, right? So what happens in the limiting case when you have like hundreds or thousands of these together? Is it exactly the same picture that is like multidimensional?
You mean this? Okay, that's the exact next thing I'm talking about. Okay, so if you have one input, you can get different sigmoids depending on location. If you had two inputs, you could get a 2D sigmoid. What's the limit of n? See? Good, that was a perfect time for that question. Yeah, what goes here? Like, if we have 1,000 inputs, do you have a 1,000-dimensional sigmoid? Are you saying yes? Yes. Well, okay, here's the reason to not get too excited. Well, first of all, actually, the thing I'm going to leave you with before we get out of this room, which is in 20 minutes, we want to make sure that we're not, as we kind of go down this path and abstraction gone wild, we do not want to keep finding ever more complicated stuff and getting really excited about how great that is, until we have some feeling that there might be that it might have some applicability to natural computation. So I'm going to show you that next. But yes, this is a question that your just mind locks onto when you see, okay, 1D, 2D, and then I can, 3. Oh wow, brilliant. Well, so what about n? But the thing that weighs against, this being like a million and having a million-dimensional sigmoid here is that remember that as you add more inputs to a dendrite, they become closer together. And since dendrites have cables and things are smooth, and if you move something over 1 micron, it's not going to make any difference whatsoever. So you're going to reach some kind of a limit where you can't tell, you get no further benefit. You reach a diminishing return of having, or trying to describe it with a higher dimensional sigmoid. Sure, it'll be a 10-dimensional sigmoid, but it's not going to look different than the 5D sigmoid or something like that. So what is the limit? So we investigated this, and this was something with Lei Jin, my student. Where do you max out? And the way we did it was we took a dendrite. This is actually not a fake, it's like a real dendrite morphology sticking out of a pyramidal neuron, and we had a compartmental model of this. And we applied, we just spatially varied differently the same number of inputs. So we took 24 total excitatory synapses, and we activated them either in random positions, clusters of 1, and here's a few of those, and here's a few 2s, where the 2s went together, and here's some 3s, and here 4s, 6, 24 divides a lot of ways, that's why we use 24. And then we could have one where we had literally all 24 synaptic contacts at one point along the length of the dendrite. And the goal of this was, and this comes back to I want to be able to do this with a piece of paper, pencil and piece of paper. So here was the algorithm that we wanted to try to use to predict how the cell would respond to every possible one of these patterns that you could generate easily with a pencil and paper. So what would you do? What you do is you run zillions of these, okay? You run a whole bunch of these, you run a whole bunch of these, a whole bunch of these, and you collect the data. Now, then how do we represent the data? What we're going to do is we're going to take bins, little lengths of the dendrite, and how many bins are there? 1, 2, 3, 4. There's 5 bins in this case, but there could have been 2, there could have been 3, there could have been 4, and there could have been 5, and that was all that we went to. It was 5. And then we would just simply count how many fell into this bin. Okay, so if it was this case, then these all would have fallen into that bin, so I would get 7 in that bin, and none in this bin, and none in this bin, and 4 in this bin, and 3 in this bin. So we would turn a stimulation pattern into just a bin count. And we would just store that firing rate that we got from running the simulation. We run a simulation, we would get a firing rate, and then we would put that firing rate into a table that was indexed by how many fell into each of these bins, how many synapses literally just drop down into these columns and counted. Does everybody see the algorithm? I mean, what we did? So we generated tens of thousands or hundreds of thousands of examples like this. And then all we asked was, when we generated a new example, a cross-validation set, and we counted how many synapses fell into each of these bins, and we looked up into the table, and we said, well, how many spikes per second are we supposed to get from these kinds of examples on average? And it's 23 spikes per second. We could then run that simulation and compare it to 23 spikes per second and see how good a prediction we got. Okay, that's what we did. And here is the quality of those predictions. If we had 2 bins, here's what we would predict, and here's what actually happened. And we get a correlation of 0.56, R squared. By 3 bins, 0.78, 4 bins, 0.89, 5 bins, 0.94. Basically, by the time you had 5 bins, you could pretty much get a very accurate representation of what's coming out of this thing. Here's what, if you want to look at it, if you want to think of this as an asymptote. So from the point of view of spatial integration of excitatory inputs on a dendrite, this sigmoidal thing kind of gets up to, well, by the time you're already here, even here is you're getting pretty good, you're pretty much nailing down the spatial analog computing capability of that dendrite in a purely excitatory case. I keep saying that as a caveat. There's no inhibition here. But it's still interesting to know from a pure excitatory point of view. And the answer is about 5 dimensions of computational nonlinear, bona fide nonlinearity, because that's how many basis functions, essentially these bins, it takes to predict how well, or predict well, what's going to be produced by the neuron. Yes?
It looked like the bin sizes were different?
Yeah, so that's a good point. Ley tried for 5, he would try every possible 5. So he would, how do you divide up this thing into 5 bins? There's a lot of ways, right? I can't tell you what the number is. Somebody here probably can. It's some set theory thing or something. How many ways can numbers add up to this? And so he would try every possible one, and then this would be the best one. Or there would be some that are best, and that's the one that's shown. So you can predict this well with a 5, this particular 5. You could find different shaped 5 bins that wouldn't be as good.
Well, how much does it vary?
Well, if you had 4 bins that were 1 micron long, and then the 5th bin, you could. So you can't answer that. I can't give you a satisfying answer to that question. But we have to admit that there would sometimes be slight different binning kind of things that would produce similar results. So we don't really know exactly what the pattern was. Why it seemed like it was better to have kind of finer representation in the middle and maybe less representation at the end. There may be a biophysical reason why that's true, but we didn't get to the bottom of that.
So just to clarify, this is fitting the compartmental simulation.
It's a way that I can. This is the pencil and paper thing that I said. You specify. I'm going to put the synapses here, and I'm going to activate them at this rate. And then I'm going to just count how much excitation goes into the 5 bins, and I'm going to look it up in my table on my piece of paper, and I'm going to say, you're going to get 40 spikes per second. And that's my prediction. And then we do the simulation, and this is what comes out. And that's how well I can do. I can predict the compartmental model. And we know the compartmental model is a pretty good predictor of the neuron. So I would say I'm very pleased with this. It means that pencil and paper model, this is what makes me think it has a shot of working. Because we know that if I could predict the excitatory the response to what's coming out of one dendrite, and I know that the dendrites kind of add the results of the cell body, I'm clawing my way to having a model to predict the whole thing. Now, we have to include inhibition in this, and we have not, and that's going to be more complicated, so who knows how well that's going to actually work. Okay. I'm going to skip. You know what it is? I'm going to just say this so that you can enjoy this. But it's something that you should, that really just troubles you. Which is, this is what happens in a real neuron when it starts getting excited. Here, a current is being injected in here. And do you see the neuron fires an action potential? Do you see that the entire basal dendritic tree just lit up like a, firecracker or whatever? So in that moment when an action potential is generated, the voltage signal in the entire dendritic tree is it's like hitting the clear button. Every branch, every place on every branch, it just gets set to the same voltage at the same time. So it's like a voltage reset button on the dendritic tree. This is very problematic. So this could be happening if the cell is firing 50 times a second. That means 50 times a second. It's like going click, 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 click to the voltage signal in the dendritic tree 50 times a second.
That vertical is the axon going up?
This is voltage at the cell body. Oh, this is the apical dendrite of a pyramidal neuron. I'm really calling your attention. I'm sorry. I meant to call your attention to this part of the basal tree because these dendrites are all kind of the same and they contain like half of the synapses of the neuron. This is a different part of the neuron which I'm actually not talking about, but I should have said that. But I'm saying if you just sort of look at this thing, kind of just it flashes, it's just sort of a slow flash. So that's a brief annihilation of the voltage of any voltage information in the dendritic tree many times a second. How could this not destroy the computation going on, which is voltage dependent? And supposed to be having separate intricate voltage things going on in different dendrites.
Maybe it's the clock.
Well, okay, now you're asking what is it for, but I'm asking why doesn't it destroy everything?
There is a clock pulse on the computer that...
Right, no, that would be why to have it, but I'm saying, okay, I'm saying why does this not destroy the independence and the sensitive voltage dependent processing of the dendrites? I'm going to answer that in a second.
I mean, isn't this a linearly superimposed signal? So the underlying signal is still there. It's just sort of everything raised and then drops down again?
Well, I wouldn't call it linear. And saying that everything is still there is an interesting thing to say. And that's sort of, there's a spirit in which that's true. So I'm gonna just show you some experiments that we did that make a point. Well, let me tell you what we did, and then I'll tell you quickly the answer. Here, I'm kind of breaking what it is I said I would do, but I think it's kind of interesting. So we could put using a simulation, and we used a simulation to study this issue. We could put two inputs here. Remember that would generate, because of their separation and whatever, that would generate this sigmoid or this input-output curve. And we could put two here, and that would generate a different input-output curve because these have different positions and it's on a different dendrite. And what we would do is we would just activate this one branch by itself, and we would then record what happens at the cell body, and that's what this graph is. Branch B's graph, and this is branch A's graph, separately from just doing it by itself. And then the question is, as we put this many synapses on branch B here and proximal these two locations, and we put this many, some point here at these two locations, and then we use that setting to predict what this branch is going to put out. We grab that number, and then we grab the number from here. Can we just add those two numbers, and that's what the cell body is going to do? So that's what we did. And, oops. Okay. And, well, that's kind of the logic of this. Actually here, okay, unfortunately, I'm showing you an example where we just had one. The input for the blue was this sigmoid, and the response for the red one was this sigmoid, and we would just put a combination of two inputs. We would read how much was on the blue, we'd read that number off. How much was on the red, we'd read that number off. We'd add the two numbers and put it into the somatic FI curve and say, that's our prediction. And when we did that, we would get predicted responses like this. The actual responses were like this, and the error was like that. It was virtually perfect. And look, this cell is firing at 100 spikes per second. So for whatever reason, it's an incredibly sort of linear summing device from the point of view of summing the effects of the different branches, despite the fact that it's firing like crazy, all the while that there's nonlinear voltage-dependent stuff going on in the dendrites. And we did the same thing with two inputs. This is where we analyzed it. And I'm not going to tell you about this. I'll tell you the result. Actually, I'll just show you here because I think Wolfgang, maybe you were the one who said it, or somebody. Oh, you said it just leaves things alone. Right. The basic reason is that these underlying waveforms that are produced by the synapses that have to do with these NMDA things are relatively slow time course events, and they are relatively briefly interrupted by these action potentials. The action potential comes and goes, and then the condition of the state of that dendrite goes back to just what it was a millisecond before. And so these perturbations are very, very brief, and they do not change the overall contour of the currents that are injected from the synapses. And so if I were to, and I'm going to skip this, I'm going to skip that, it's kind of interesting, but I'm just going to show you these predictions. If we had two inputs at two locations on two different dendrites producing these two separate things, if we use that simple algorithm, we just pretend what's coming out of this one by independently, what's coming out of this one independently. We ran hundreds of thousands of cases, and we made the prediction by adding those two numbers together, putting it into the somatic IV curve, reading off a prediction, and here were the actuals and here were the predictions. R squared equals 0.9999. So despite this apparent crazy synchronizer of the cell body, it actually is helping to, what we showed was it actually helps to, because the cell is firing action potentials, and action potentials are very stereotyped, and they return the voltage back to a stereotyped place, they help isolate the voltage changes in the different dendrites from each other and create a more independent subunits. So we have a paper about this if you're interested. And anyway, so that reassures us that the two-layer model is still a good workhorse, because even when you have nonlinear things going on in the subunits, the second layer appears to still behave rather simply. Yes?
Now you seem to say that if you have these spikes and the voltage sort of propagates into the dendrite, that helps in this linear superposition of the contributions of the different compartments. So in your simulation, can you sort of turn off the spikes and show that then the superposition is less linear?
Oh God, that was a perfect question. That's what this graph is that I skipped. It's basically showing that what we don't want to have is that voltages, currents coming out of one dendrite affect the input-output curve of the other dendrite. That's what we don't want, because that's a crosstalk thing. And that means that we can't tell what this dendrite's going to spit out. It depends on what's going on in other dendrites. Then they're no longer independent. So this is showing the input-output curve of a dendrite being affected by what's coming from the cell body. And we don't want it to be affected. So what happens is, when the cell is at rest, then this is the input-output curve of that dendrite. If the cell is depolarized at the cell body by somebody else, then that guy's input-output curve shifts a lot. It shifts from here to here, which is a huge threshold change. But then the minute the cell starts to fire action potentials, 1 spike per second, 50 spikes per second, 100 spikes per second, the input-output curve is just locked down. And it's no longer affected by how much the cell body is being activated by other guys. It's kind of amazing. Yes, Marie?
Wouldn't the membrane time constant spread out the back-propagating action potential in time?
Yeah, there's some of that. You know, an action potential will become a little bit fatter as it moves out of dendrite. This has been, people have done back-propagating action. But it's still like a 1 millisecond long thing, you know, a couple milliseconds at the most. I mean, I can show you, I mean, at least in a simulation. And there are studies of this. There's a little bit more to it. You know, there's some other sort of second-order effects that are going on here that I didn't really have time to. I think I'm going to just have to skip to the end here because we've run out of time. But so there's a section which that I want to get to, or just the last slide I want to get to. I was going to show you an application. I mean, I was going to show you where sigmoids come from, and then actually or where they are found. We found a place where I'll just show you this one picture and just tell you, and then maybe we have time to talk later. We measured something in natural images, a nonlinear interaction between different places in natural images in the context of contour detection in natural images. We actually collected natural images, we labeled contours, we looked at interactions between center and flanker inputs, and we found that the interaction between them in order to compute contour probability in natural images, something a neuron needs to do to detect contours in natural images, is a two-dimensional sigmoidal function. And if you were to take inputs, these values, and put them onto a dendrite at two different locations, it would produce that function. And that function, the black frame is the same in both graphs. So this is what the dendrite likes to do and does naturally, and based on its biophysics. That's a natural visual computation having to do with contour detection. It's a multidimensional sigmoidal shape. So we have one example in our hands of where with the first place we looked, you see these types of relationships between sort of a primary variable and a modulatory variable. They crop up. So we think that the hardware is at exactly the right spot it needs to be to help perform these computations. Okay, and with that, I'm going to go to my last slide. Oh, yeah, look, I had Jeff Lichtman's slide in here. Oh, God, never mind. Oh, here it is. Yeah, so what did we learn? This has to do with, like, when you look at stuff in the brain, what should you see? And one thing you should see is, this is a neuron. This reminds me of those CNN commercials. This is an apple. This is not a banana. Have you seen those? Oh, you haven't? Oh, you don't watch enough TV. But, so that is a neuron, the way that we think about a neuron. It's as elaborate and complicated as it is, it really is like a point neuron, like a conventional point neuron. And then this is not a neuron. This is a neuron. Well, I mean, I'm making a point where the thing that we used to think of neurons are is a little integrated unit that generates a local spike. It's sort of like one of these. And I mean, but you wouldn't have known it. And then this is actually, in fact, this thing, because of these kind of spatial effects, in a way is a more complicated nonlinear processor than that thing. So that's one kind of interesting thing. And actually what you would call that is you would say that is 30 complicated neurons with a shared learning signal. So I didn't talk about this, but that backpropagating spike that comes out, a lot of people think that that's to help to sort of inform for plasticity in the dendrites. It kind of creates a synchronized plasticity signal so that these dendrites are all learning under the same signal.
Sorry, which backpropagating spike?
When the soma fires, it sends the voltage out into the dendritic tree. Everybody gets it. Every synapse gets to feel it. So they all know that the soma spiked. And so if you want to have an STTP rule or some kind of a rule, they get broadcast that signal. And they all get the same one. So it's different to group a dendrite together into this neuron versus the next neuron over, which gets a different signal. And then this is not an interconnection. It's a synapse. That's an interconnection. So when you want to think about the functional interconnections between neurons, this notion that it's just an excitatory or inhibitory weight is overly simple, because the way that neurons probably like to interact with each other, and probably a more useful abstraction of what an interconnection looks like in the cortex, is that it's something that creates a conjoint excitatory inhibitory effect distributed possibly non-uniformly across the dendritic trees of a cell that's being targeted. So it's like a higher dimensional interconnection weight or interconnection. It's like a multidimensional interconnection in the same way that these are like multidimensional sigmoids. So we're getting, it's an odd computer and it's an exciting computer to be studying, and we're still, I mean, there's so much, just it's mind-boggling. But anyway, I hope you get some ideas from this and help you in your own work. Thank you.
So what is the mechanism for filtering out the back APs?
For filtering?
So this backpropagating action potential that you mentioned, so how does the cell filter them out as like opposed to the inputs that are actually coming to the dendrite?
Well, the way that we would say that is that we studied cases where there was sort of a longer time course. There's a train of inputs being delivered to synapses over a period of time, which is long compared to the brief period that an action potential lasts, which is on the order of a millisecond. So if a cell's receiving, when you fixate an object in vision, typically a fixation lasts 300 milliseconds. So maybe that's a reasonable amount of time, a third of a second or a fraction of a second, where your brain is integrating the signal from something. That's long compared to the period of an action potential, which is like a millisecond too. And so we think of what's happening in the dendrite as being on a slow time course, and these interruptions as being very, very brief. And so they don't mess with the voltage signal for very long. They don't alter the currents that those synapses would inject very much. And that's what kind of helps. That's why they don't perturb very much the computations going on that are dependent on the voltage dependence of the NMDA channel. Yeah.
You were talking about the backpropagating action potential.
Yes, and that's what I was.
Those are calcium I was talking about calcium spikes.
Oh, you're talking about.
So actually, what clarifies it's a little unclear to me because the calcium signal can last a very long time.
Right. So what we've been talking about, what I've been focusing on, are these thin dendrites that like perisomatic thin dendrites or oblique dendrites on an apical. If you activate the apical tuft, you can get calcium spikes, which is a different type of a biophysical mechanism, which are, yes, they can be much longer lasting. But they seem to, that's not the main, blocking calcium channels in these dendrites does not make NMDA spikes go away. It can just changes a little bit, but it's not, they're not a big current carrier in dendrites, the thin dendrites.
Yes.
In this pursuit of abstraction gone wild, would you say there's anything about that pyramidal cell that could not be reduced to a network of simple neurons, say point neurons? Replacing each dendrite, for example, by a point neuron.
Well, so, you know, the computer scientist in everyone doesn't see any distinction between if I call this a point neuron and then they all add together and then this whole thing is just a big sum of point neurons, it's just two layers of a network, and I don't care what you call a neuron or not. I could draw a circle around this many units in a diagram and say, okay, that's what a real neuron actually corresponds to, but we wouldn't care from a computational point of view. But so the only thing I think, well, there's the way that the different subunits of the neuron are more related to each other inside a neuron than they would be between neurons is kind of an answer to your question. And I can't make a super compelling, I said one idea, which is that they certainly share a learning signal, a timed learning signal, which they share that is private to that neuron. They also share the modulatory state that you know, there's a modulatory state that is sort of affected by the apical tuft, which will be shared between them too. So neurons actually have a meaning. I mean, you know, when you look in the visual cortex and you record from a neuron, it typically has a receptive field that's somehow interpretable. It's responds to some kinds of objects, and you know, and somehow things that want to modulate, like if you want to attend to a certain object, you want to attend to a certain location, or you want to attend to a certain, you know, you're looking for a vertical. There's ways in which you might want to modulate all of the subunits of a neuron together and differently from the next neuron over. So, but yes, I think we're not going to be blown away by the ways that, you know, your question, the spirit of your question is exactly correct. How much should we care that a bunch of different subunits is grouped within one neuron versus we just, you know, but I think the groupings have to do with plasticity and have to do with modulation.
I was just, so getting back to the five-dimensional thing, so if I understood it correctly, with the right sigmoid, like within some length of dendrite, you don't need more than 5 synapses. So like you kind of need just some like sparse you just need some sparse synaptic connections. Is that what you see?
No, I didn't say that. I was just saying that, you know, a typical dendrite, like one of these dendrites, might be covered with a couple hundred contacts, and they'll be kind of, you know, all along. I mean, you saw those pictures that Jeff had yesterday. Now, we're just saying that you could, if you want to know what the dendrite's going to do as it gets stimulated, and there's some pattern of excitation coming in along its length, you can sort of think of it as being binned, and okay, there's the ones that are kind of close to the soma, there's the ones that are here, there's the ones that are here. How much excitation is coming at several different sort of lengths along the dendrite, and just by knowing that kind of sum of, you know, like binning of how much stuff fell here, 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 and here, you could take those 5 numbers, and that allows you to predict what that dendrite is going to put out. So you look it up in a table. You said in all the cases in which you had this much excitation at these 5 places, here's the average firing rate that was produced in those cases, and that's your best guess. So you guess that, and then you find that you actually get something very close to that. It's a good predictor.
I think over there.
Oh, Bruno? Or
yeah, so along the lines of Brian's question about, you know, how easily can you replace this with a bunch of point neurons. So at the beginning, you introduced this two-dimensional landscape of morphology and dynamics. So it seems like we've been talking a lot about morphology, but, you know, so when we think about the dynamics, things like coincidence detection and that so forth might come into play.
Yeah, that's a very good point. I've been kind of focusing this entire time on spatial processing. So, well, Bruno, you just said something. There's 2 things that you might add. You said a network of point neurons. Okay, we've already kind of established that these types of dendrites don't look like point neurons, but they could still be a network of neurons that are a little bit more complicated, that do have spatial processing in them. But they'll have temporal processing too. That's an additional complication. But then we're just going to get to a point where these things can be interesting analog spatial processors and interesting analog temporal processors. It just generalizes, it just makes the subunit more complicated, but which is fine. We've already made it more complicated. We can make it more complicated by adding time in there. But that's a good question. What does it become when we add time? And that opens up a huge thing. But we still may have a bunch of more complicated units that sum within a neuron. That may still be a valid abstraction. And as far as we can tell, it sort of is. So that's kind of the answer to your question, is maybe until we find a really bad breaking of the prediction that the effects of these things kind of combine very simply in a linear way, then maybe it's okay to just call them a bunch of units that share a training signal. I don't know.
So in the middle of your talk, I thought, well, great, even though the neuron is complicated, I can use a standard neural network to simulate everything, because that's the standard neural network. But then you said they share a learning signal. I mean, they also funnel their information through one axon, right? That's the other thing that's...
Yeah, that's true. Their output sum.
So I was wondering whether this now, I mean, does this have a particular function? I mean, do you see particular learning problems where this would be useful as compared to a more general network? Or is this just a cheap way of getting sort of a lot of computational units that are not quite as universal as a network, but it's much cheaper, sort of biological to get that? So is there a function for this, or is this just a cheap way of getting a lot of units?
That's a good question, and I wouldn't try to give you a general answer, and then if there's a general answer in the cortex, it may not be the same as in the striatum, or wherever else we look. But one of the ways that this relates to things that we may have thought about elsewhere, by we, I mean all of us, is to think about the multiple subunits of a neuron. What might they be? And a lot of people who've been interested in vision and who can remember back to Fukushima and so on, we all like the idea that a neuron pools together subunits that represent shifted or translated or rotated or something. Viewpoint varying versions of the same thing could be pooled together inside a unit and create a more invariant receptive field, a complex cell, if I may say it that way. And so we're very used to that idea. And David Hubel said it, that you could have dendrites being the subunits of a more like a complex cell. And so as you move through the visual system, there is a progression towards larger and larger pools of things that are more and more complex. And I believe that that's going to turn into be that it's the dendrites that are the pools. The collections of dendrites within a neuron are the pools. I mean, it's not a huge leap from this. It's something that we've always thought. So that's one reason why you might be building pools inside neurons, because they represent the same thing under sort of, but they're helping to absorb variations in the stimulus by making the neuron respond to all of them. We have a couple people over here.
Yeah, okay.
Yes.
So you talked about the dynamics you talked about what happens in the branches near the soma. What about the one that goes all the way up?
Yeah, I didn't talk about that. People have done work on this, and they believe that this is kind of a global modulator of the neuron, where it kind of sets the gain for the whole neuron, like all the subunits. And Matthew Larkham is one of the main people who's kind of done the experimental work behind that. Walter Sen had a modeling paper to look, was it a modeling paper? No, I don't think it's been so much of a modeling paper, but Walter Sen and Larkham did a paper on that. And that's kind of, I accept that. I think we should break, right, Bruno? Because of lunch?
Great. We'll have lunch, and then thanks very much, Bart. We'll resume at 2 o'clock with